welcome to the Explainer. I am so thrilled you're joining me today because honestly, we are going on a truly fascinating journey. We're going to explore the literal physics of how we see the microscopic universe. We're talking about the hidden structures of life itself, the fundamental building blocks that make up you, me, and literally everything around us. But to actually see the machinery operating inside your own cells, well, we have to overcome some incredible physical barriers to do that. So get ready, focus in, and let's uncover exactly how science makes the invisible visible. Right off the bat, we're faced with this massive, completely thrilling scientific mystery. How exactly do scientists look at things that are smaller than light itself? We always think of light as the ultimate tool for seeing. You flip on a lamp in a dark room and boom, suddenly everything is revealed. But light actually has physical limits. It has a size, a measurable wavelength. And when the things you want to study are actually smaller than that very wave of light, what do? This isn't just some fun piece of trivia. It's a profound physical roadblock that scientists had to completely engineer their way around. Okay, let's dive into this by clearing up a very common misconception. When we talk about microscopes, people almost immediately think of magnification, right? Making things bigger. But magnification is simply the observed size divided by the actual size. It's just blowing an image up. The problem is, making a blurry blob bigger just gives you a giant blurry blob. What we really truly care about here is resolution. Resolution is the absolute minimum distance between two objects where they can still be seen as two distinct, different objects. It's all about crispness, clarity, and the actual ability to distinguish tiny details. Now, when scientists are looking through these lenses, they have to actually measure these tiny, highly resolved structures. And obviously, you can't just stick a regular wooden ruler under a microscope lens. Instead, they use very specific microscopic rulers. First, you have the eyepiece graticule, which is basically a small scale placed right inside the eyepiece itself. But to know what those little marks actually represent, scientists have to calibrate it using a stage micrometer. This is a small, incredibly accurately drawn scale with known dimensions that is engraved right onto a microscopic slide. You compare the tool in your eye to the physical ruler on the stage, and suddenly you can measure the entire microscopic world. But here's the kicker. Even with absolutely perfect lenses and perfect measuring tools, a standard light microscope eventually hits a hard, immovable wall. And that wall is exactly 200 nanometers. That is the absolute maximum resolution of a standard light microscope. Meaning, if two tiny structures inside a cell are 199 nanometers apart, a light microscope is just going to blur them together into one single object. We are completely blocked from zooming in any further using standard visible light. And here is exactly why that happens. The fundamental rule of physics holding us back is that to be visible, an object must be at least half the size of the electromagnetic radiation's wavelength. Just let that sink in for a second. Visible light is part of the electromagnetic spectrum. Violet light has a wavelength of 400 nanometers, and red light is up at 700 nanometers. So if we use the absolute tightest visible wave we have, violet light at 400 nanometers, the math dictates we can only resolve objects down to half of that. Half of 400 is 200. And boom, there is our 200 nanometer limit. Physics literally says, nope, no further. So this forces a really critical question. What if an object we desperately need to study, like a ribosome, the tiny protein factories in our cells, is only 25 nanometers wide? This is a total mathematical paradox. 25 nanometers is way, way smaller than half the wavelength of even the tightest violet light. If we try to bounce visible light off a 25 nanometer ribosome, the light wave essentially just washes right over it. The information is completely lost. And if the size issue wasn't bad enough, we also have a transparency problem. If the microscopic object you're trying to look at is transparent, light waves will literally just pass straight through it. It won't interact with the light in any way that creates an image for our eyes to see. So for incredibly tiny, transparent cellular structures, visible light is utterly useless. We need a new hero. Enter the free electron. So when a metal becomes extremely hot, it gains so much energy that it actually loses some of its electrons from their orbit. These free electrons behave like electromagnetic radiation, but they come with what I like to call physics superpowers. First, they have an extremely short wavelength, far, far shorter than visible light. And second, unlike light, electrons possess a negative charge. This is absolutely crucial because it means instead of using glass lenses, we can actually focus this beam of electrons using powerful electromagnets. And this brilliantly illustrates the kind of power we're talking about, 0.5 nanometers. By harnessing that incredibly short wavelength, electron microscopes completely shatter the 200 nanometer limit of visible light. We go from a limit of 200 nanometers all the way down to a breathtaking zero point 0.5 nanometers. It is an astronomical upgrade in our ability to resolve the universe.
Now, how do we actually put these electrons to work? Well, we generally split this incredible technology into two distinct methods. You've got transmission electron microscopes, or TEM, and scanning electron microscopes, or SEM. With TEM, the electron beam is fired directly through the specimen, which gives us a highly detailed 2D view of internal structures. SEM, on the other hand, fires the beam onto the specimen and it bounces off. It reflects. The scanning method gives us stunning 3D images of surface structures, though it is at a slightly lower resolution compared to TEM.
When we break it all down, the core difference is honestly brilliant. It really just comes down to the difference between looking through an object versus looking at its surface. TEM captures the electrons that actually make it right through, while SEM observes only the reflected beam to scan the outside topography. But wait, humans can't see electrons. If you stick your eye over an electron beam, you aren't going to see a picture, right? So how do we actually view the final image? The technology projects the electron beam onto a fluorescent screen. Wherever those electrons hit the screen, it shines brightly. Areas where electrons were blocked by the denser parts of the specimen remain dark. This creates a highly detailed overall black and white picture of the microscopic world. So the really crucial takeaway here is that electron microscopes only produce black and white images. If you've ever flipped through a biology textbook or seen an article online with vibrant red blood cells or neon green viruses, you are looking at what's called a false color image. This is simply an image created by taking that standard black and white electron picture and adding colors to it later using a computer. It makes the structures easier to differentiate, and frankly, it just looks way cooler. But, of course, this incredible super resolution comes with a massive technological catch. You can't just toss a bug under an electron microscope on your desk. The entire interior of the microscope must be a complete and total vacuum. Why? Well, because if there's any air inside the chamber at all, those tiny charged electrons will crash right into the air molecules. They'll scatter everywhere, completely destroying your beam and leaving you with a totally useless, low-resolution image. Let's move on and see how this builds into a pretty grim biological reality. Think about it. Step 1. In a vacuum chamber, the pressure drops so incredibly low that water literally boils at room temperature. Because of that, step 2, any living material must be completely dehydrated first, which leads to our tragic step 3. You can only ever examine dead, non-living material in an electron microscope. The literal physics that allow us to see life at this scale actually require us to end it first. But, despite that limitation, the payoff of this technology is monumental. Bypassing the limits of visible light allows scientists to observe what is known as the ultrastructure. This is the incredibly fine, detailed internal structure of a cell that can exclusively be shown by an electron microscope. It's the hidden architecture of biology that light could just never, ever reveal. This was the exact moment biological science just leaped forward. Suddenly, all these specific, tiny physical structures were jumping out at researchers, things that were totally invisible before the electron microscope came along. Let's break down exactly what we discover when we finally look at this ultrastructure. First, the cell's surface membrane. We can finally see it functioning as a partially permeable barrier that actively controls the exchange of materials in and out of the cell. Amazingly, the electron microscope reveals this membrane actually has 3 distinct layers, 2 dark layers sandwiching a pale, narrow interior layer. And we can also clearly see structures like microvilli. These are tiny, finger-like extensions on epithelial cells that brilliantly increase the surface area of the cell to make absorption much more effective, like what's happening right now in your own intestines. We aren't just seeing blurry blobs anymore, we are seeing the literal machinery of life at work. We started with a physical limit, didn't we? A wall of visible light that hid the deepest secrets of biology from us. But by understanding the physics of the electron, we totally bypassed that wall, illuminating a microscopic universe all the way down to 0.5 nanometers. But it leaves us with an incredible thought to ponder. Now that we can map the complex, 3-layered ultrastructure of a dead cell inside a vacuum, what invisible microscopic frontiers will human ingenuity conquer next? How are we going to innovate our way around the next unbreakable rule of physics? Thank you so much for exploring the invisible with me today. Keep asking those big questions, keep looking deeper, and I will catch you on the next explainer.